Okay. Uh, Next chapter we are having is chapter number 4. Now from chapter number 4 till chapter number 8, we will discuss all the topic which we have discussed in the classification, means the kingdoms. Okay. Going to discuss the protoctista, we are going to discuss the fungus, we are going to discuss the plantae, we are going to discuss the animals. So all these chapters from chapter number 4 to chapter number 8 are regarding classification. We will discuss all the classification over here. Okay, let's start from chapter number 4, that is viruses, bacteria and cyanobacteria. Okay, माइक किसी का ऑन है तो काइंडली अपने माइक बंद कर दो, लेक्चर की डिस्टर्बेंस नहीं होनी चाहिए, kindly all of you. So let's start with the topic number 1, that is viruses. Let's see what viruses are. First of all, the introduction to virus. Viruses basically derive from a Latin word that is viron, and viron means poison. Okay, so as the word name suggests, poison, it means that they are dangerous organisms by any means. They cannot help you anything. They are going to give you harms. Okay, they are the simplest, smallest and primitive living things. Simplest, smallest and the most living thing, the most lived living thing. You can say that they are most in number. Everywhere they are present. Okay, viruses were discovered in the year 1892 by a Russian biologist. His name was Ivanovsky. And where he discovered these viruses, he described the viruses in the leaves of tobacco, and he called them as TMV virus, that is tobacco mosaic virus. It was causing diseases in the tobacco leaves. The disease was known as TMV disease. So in the year 1892, the scientists were basically able to find out little bit about viruses. Okay, it was not, but we knew about bacteria before that. So as the scientists saw that these organisms are not bacteria and they cannot be seen with the light microscope, but they are organisms which are smaller than bacteria, so they gave the name virus to them. Okay, virus is derived from viron, which means poison. So Ivanovsky was a scientist who initially worked on that. After that, when electron microscope was discovered in the year 1935, we became able to find out more about viruses. Okay, so it was only when the electron microscope was present. After that, we study the structure of the virus, that what the virus are made up of, how they infect the living organisms and other things, and what are the properties of the virus. They were discussed after the discovery of the electron microscope. So this is a small and brief introduction regarding the virus. Okay, again, viruses are, the word virus is derived from a Latin word that is viron, and viron means poison. They are the simplest, smallest and most primitive living things. Okay, viruses were discovered in the year 1892 by a Russian biologist. His name was Ivanovsky, and where he described it, he discovered them in the tobacco leaves where they was causing a disease. The name of the disease was put as TMV disease, tobacco mosaic virus disease. And then in the year 1935, we discovered the electron microscope, which make it possible that we can study the structure of the virus, we can study the properties of the virus, we can study the infection caused by these viruses. So this was all about the discovery of virus. Let's see the next. What are the characteristics of a virus? Number 1, they are parasites. Now, what are parasites? Parasites are those organisms who are living in their host. They will harm their host, and in return, they will harm their host. They're called parasites. Okay, so viruses are basically non-cellular obligate parasites made up of cells. You will not see virus anywhere in the classification because they are not made up of cells. They are non-cellular. Okay, and they are obligate parasites. Obligate parasites mean that they only survive in their host body. They cannot survive outside the host body. So they are known as obligate parasite, and they are going to harm you obligatory. Whenever the virus comes to your body, what is the function of the virus in your body? To harm you, because they are obligate parasites. Okay, they cannot produce outside the host body. They can only inside the body of the outside the host body. They are completely inactive. There is no work of the virus outside the host body. Now, what is the structure of the virus made up of? The structure of virus is made up of protein coat and a nucleic acid core. Okay, the protein coat is normally known as capsid, and the nucleic acid core may be DNA or maybe RNA. They can be deoxyribonucleic acid or they can be ribonucleic acid. So one of the two nucleic acid will be present in them, and the protein coat will be present in them. Okay, that is the structure of the virus. What is the shape of the virus? You will see that virus appears in different forms. There can be small rods, there can be threadfold shape, they can be polyhedral in shape, they can be like spheres, and many more shapes are present of the viruses. So viruses have many shapes. They do not have any particular shape. They have many shapes. Okay. Then what is the size of the virus? They range in size. Okay. Normally the smallest virus is of 20 nanometer. Nanometer is a negligible particle. -9 is the power of nanometer. So the smallest virus is of 20 nanometer, and they can range up to 250 nanometer. Into 220 to 250 nanometer. That is the size of the virus. Let's see reproduction of the virus. Virus basically not have any, you can say that particularly any sexual or asexual reproduction present in them, but they can produce replications. They can make their copies. Okay, and the copies are made by the enzymes which are present in their body. They inject that enzyme into the host body for protein synthesis and then gene replication. So protein is synthesized inside the host body and gene is replicated, DNA or RNA is replicated, and after that the body of the virus is made. So they can only produce within the host body. Okay, outside the host body they are completely inactive. They cannot do anything when they are outside the host body, but as it enters the host body, it is going to produce, and after that it is going to harm the host. Okay, let's see the genes present in the virus. Virus is basically a complex structure. Okay, sometimes it is more complex and sometimes it is simpler. So if there is a simple virus, you will see 4 genes in them, 4 characters in them, and if they are the complex virus, you will see up to 200 genes, maybe 5, maybe 10, maybe 20, maybe 50, maybe 100, maybe 150, or maybe 200. So up to 200 genes can be present in a virus. What is virulent? Viruses are virulent. Virulent mean they can remain stable inside the host body for longer time, and they can destroy the host body. It is virulent mean poisonous. Okay, so they live inside the host body, they survive over there for longer time, and then after that they destroy the host body. Slowly and gradually the virus destroys the host body. Okay, so before we move toward the next topic, if you have any question from this topic, you can ask so that we can move to the next. RNA. RNA means ribose nucleic acid. Ribose nucleic acid. Barira, I will send the recorded lectures as well. Protein coat. Protein coat is known as capsid, which is made up of proteins. We will study the structures. You will see that how the protein is formed and how the other DNA is formed.
Sir, can you explain again parasite?
Parasite are those organisms who cannot prepare their own food. They live in their host body. They get their food from the host body, and in return, they give harm to the host body.
Sir, are viruses living organisms?
Viruses, they are debatable. We will discuss that.
Sir, the TMV you said na, the tobacco mosaic virus. What is mosaic?
It is basically a structure. Mosaic is basically a zigzag structure. So they were
Okay.
present in a mosaic way. So that was basically called a mosaic virus.
Mosaic.
Reproduction, the virus reproduce in very simple way. They go into the body of the host, into the cell of the host, and after that, in the host body, they replicate. Replicate means they make their carbon copies by attaching to the proteins and DNA of the host. Nucleic acid core means the central part. The central part is the nucleic acid. We will discuss in the next topic. Virulent means poisonous. Okay. Virus can only reproduce, yes, virus can only reproduce inside the host body, and when they are inside your body, they will harm you. Outside the body, they cannot do anything. They are inactive.
Sir, what is virulent?
Virulent का मतलब होता है poisonous.
Sir, how the scientists knew that they were not bacteria and there were other types of
They knew because they were smaller than bacteria in size. They knew the size of bacteria. So they were smaller than bacteria because they were not bacteria. Bacteria were bigger. They could see them with a microscope. But the viruses couldn't be seen with light microscope. Inactive means that they do not perform any activity outside the body. When they are inside the body, then they perform their activities. $200 grams. Where I told you about the weight of the virus. Anam, kindly turn off to your mic. Anam Wahid.
Okay, sir.
I have never taught anything about the weight of the virus, beta. Neither I have weighted it. It's a microorganism, negligible mass. How can you weight that? Okay. So now we are moving to the next topic, that is the structure of the virus. Okay. If you have any question, you can ask them later on. Okay, this was about some of the properties. Okay, at least in exam you could be asked 4 or 5 properties. You should remember 5 properties out of these 7. 5 properties you should remember about the viruses. Let's see the structure of a virus. First of all, the appearance. So we discussed in the structure as well and the properties as well. That they may appear like rods, they may appear like pairs or circles. Okay, their body is made up of nucleic acid. The simplest virus contain a single molecule of only 4 genes, while it can be up to 200 genes. We discussed that as well. That is a nucleic acid. So nucleic acid can be DNA or it can be RNA. Some of the viruses have DNA, some of the viruses have RNA. Okay, so like for example, today we are having coronavirus. So coronavirus is having RNA. They have RNA nucleic acid in them. Okay, like for example, we are having cough virus, or we are having a flu virus, or we are having influenza, or we are having polio virus. So some of them are having DNA, the other of them are having RNA. So they may be having 4 characters, or they may be having 200 characters. The one who is having more characters will be more dangerous. Okay, then we are coming toward the capsid. Capsid is the protein coat. The protein coat in which the nucleic acid is surrounded. The nucleic acid is present in the center, and around the nucleic acid we are having a protein coat present. That protein coat is known as capsid, and the capsid is made up of protein subunits, smaller units of protein. Those smaller units of proteins are known as capsomers. So the capsid is the protein coat in which the nucleic acid is enclosed, and the capsid is made up of very small units of proteins. Those small units of proteins are known as capsomers. Okay, let's see the next. Viral envelopes. Now envelopes are not present in all the viruses, but they are present in some viruses. It is an accessory structure which is normally present above the capsid, above the protein coat. And the viruses who have the viral envelope, they are more dangerous, they are more poisonous to their host. If the virus do not have envelope, the virus who do not have the viral envelope in them, they are not that much dangerous. The influenza virus is not that much dangerous because it do not have the envelope. But if we go towards the coronavirus, we go toward the Ebola virus, we go toward the other viruses, the Congo viruses, they have viral envelopes in them. So they are extremely dangerous. They are very dangerous viruses. They're gonna infect the body of the host very, very badly. Okay, then they the tails are complex structure. In the complex virus, the tails are present. With the help of the tail, whenever they want to attach themselves to a host, they are going to do what? They are going to attach themselves with their tail. And the whole virus is not going inside the body. Okay, the whole virus is not going inside the body. And you can say the whole virus is not entering to the cell. Only their genes are entering to the cell. So when they inject their tails, with the help of the tails, they inject the DNA or RNA into the body of the host, and after that they infect the host body. Okay, let's see the diagram of a tadpole virus. This is a bacteriophage tadpole virus. Now you can see that the outside coat is the capsid, and you can see in the capsid these small units are known as capsomers, and inside the capsid there is a DNA molecule or RNA molecule may be present. Either it is DNA or either it is RNA. It is present inside the capsid. There we are having a tail. Okay, the tail is basically protected again by, and it is made from the, it is made up of protein. Again, you can see the smaller units of protein in that. The capsomers can be seen over there. And then they have the tail fibers. You can see these tail fibers. The virus do what? The virus is basically going to attach itself with these tail fibers to its host. And when they are attached to its host, they are going to inside the body of the host. It's just like an injection where we are injecting something to the body. So the virus injects its DNA and RNA into the body of the host. When this DNA goes into the body of the host, then this DNA is going to be host that replicates itself. So it takes the control of the host body. Okay, it takes the control of the host body. Slowly and gradually, it takes the control of the host body. So this is the structure of the virus. Again, if you have any question, now you can come and ask your questions.
Sir, where is the envelope in this picture?
Where is the envelope in this picture? You can see the outer layer, this layer, this outer layer. This is the envelope. This outer layer is the envelope. Okay, this outer layer is the envelope. Yes. Any other? Capsomers are the units of protein. Arisha, capsomers are the small units of protein from which capsid is made. Polyhedral mean which have multiple heads. Poly mean many. Hedral mean sides. Have many sides. Yes, the viruses which have the envelope, they are more dangerous as compared to the virus who do not have the envelope.
तो सर जिनके एनवेलप नहीं होते इनकी आउटर लेयर नहीं होती वायरसेस की?
जी, उनकी बाहर वाली लेयर नहीं होती है. कैप्सिड होता है, कैप्सिड के बाहर पर लेयर नहीं होती है, एनवेलप नहीं होता ना. तो when there is no envelope, they are not going to harm you more. Okay, they will harm, but they will not harm more. We will study bacteria. Firstly, we will study bacteria, then you will find out the difference between them. Viral envelope is an extra layer that is present outside some of the viruses, which make them more poisonous, more infectious. Capsomers are the units of protein, the smaller units of proteins.
Sir, above tail fibers, it was that tail sheath. What is it? Sir, above tail fibers, there was tail sheath. What is it?
Yeah, that's what I'm telling you. The tail sheath is basically made up of proteins. They have only protein present in them. It's basically a way through which they can inject their DNA.
सर वायरसेस की कोई प्रॉपर स्ट्रक्चर या उनकी शेप होती है या वो रैंडम होते हैं?
Each and every virus has its own shape. All the viruses are not same. Okay. Yes, the viral envelope is not present in all the viruses. They are present in some of the viruses. Okay, I'm showing you the diagram again. See the diagram again. You can see the diagram again here. Nucleic acid is present. The capsid is present. The capsid is made up of, you can see the capsid is made up of smaller units of protein. These smaller units are known as capsomers. Okay, then they have a tail sheath. The tail sheath is also made up of protein subunits. And then they have tail fibers. The tail fibers are the tentacles through which they can attach themselves to the host body. So when they attach themselves to the host body, they can inject their DNA to the host. To the host, the DNA is going to have a copy of itself inside the body. When they have a copy of itself inside the body of the host, then they can have control of the host body as well, and they can infect the virus, the host body.
And sir, that's how we could see the virus or bacteria in our blood by blood test.
Yeah, we can see that through electron microscope. Okay, when we go testing, so we normally test that with the help of electron microscope. Bacteria can be seen with the what we can say light microscope as well, but viruses we see with the help of electron microscope. Is it done? Anybody having any question now?
No, sir.
Okay, good. So now we have to move to the next topic. Let's see what the next topic we are having. And this was the structure of the virus. We discussed the characters of the virus. Very much important topics these are. You can be asked about the structure of the virus. The diagram is very much important. This diagram can be asked for 5 marks. Okay. So whatever I'm telling you that these things are important, keep that in mind that these are important, and in exam they will be asked. So the character of the viruses are important. The diagram of the virus is important. The structure of the virus is important. So you have to let's move to the next topic now. Are viruses living or non-living? There was a question asked from somebody, and they were saying that are viruses living or non-living? So basically, there are kind of organisms which are present on the borderline. They show some characters of living things, and they show some characters of non-living things. So again, this is an important question. This question is asked in the board exam, and they will tell you that discuss if virus is living or non-living. So you are going to tell them the living characters of the virus and the non-living characters of the virus, and then you are going to justify that they are present on the borderline of the living and the non-living organisms. They show some characters of living things, and they show some characters of characters of the virus first. Then we will discuss the non-living characters of the virus as well. So first of all, they have DNA or RNA present in them. And we know that DNA and RNA can only be present in living things. They cannot be present in non-living things. So the first character which they have is DNA or RNA, which resembles the chromosomes of the living things. So it means that they can be a living thing. So this is the first argument which we can give that viruses are living. Number two, they have the ability to reproduce, means they replicate themselves. Non-living things cannot replicate themselves, okay? Only living things can reproduce. So the viruses can reproduce. They can reproduce inside the body of the host, but they can reproduce. It means that they can be living things. The second argument. The third argument, they may undergo mutations. Mutations are basically change in the genetic material. Now you see that viruses present in different areas, they show different characters. Like for example, coronavirus present in China has a different version. Coronavirus present in Pakistan have a different version. It is present in Italy and Spain, America, they have different versions. Okay, so they have the character, they may undergo mutations. Mutations mean that they can change their genes according to the locality and according to the area. Only living things can do that thing. Non-living things cannot do it, okay? So somewhere this virus will cause a lot of problem, and some other ways they are going to cause a less problem. In some areas, it will be more violent. In other areas, it will be less virulent. It means that they show mutation. They can undergo mutations as well. Only living things. Combination. Genetic combination means that they normally can combine with other organisms and can show genetic properties. Like for example, they go to the body of the bacteria, and from there they reproduce themselves inside the body of the bacteria and can carry the genes from the bacteria as well. It means that they can show genetic recombination. This genetic recombination is only the property of living things. Non-living things cannot show this property. Okay, so these are the four things, the four properties on the basis of which we can say that viruses are living things. On the other hand, the non-living characteristics of the viruses are, number one, they are non-cellular. Okay, and how we define a cell? A cell is the basic unit of structure and function of living things. It means that any living organism present in this world must be having cells. But viruses are the organisms which do not have cells. They are particles. They are not made up of cells. Number two, they are completely inactive outside host cell. When they are not present inside the host cell, they are present outside, so they are completely inactive like dead organisms. So again, we can say that they are non-living. Number three, they may undergo crystallization. Crystals can be formed. They are particles. So they can be divided. They can be further classified. They can be further crystallized. Living thing cannot be crystallized. Only and only non-living things can be crystallized. We can make crystals, geometrical shapes from non-living things. We cannot make geometrical shapes from living things. So these are the properties on the basis of which we call that viruses are non-living thing. Now the comments are that viruses are present on the borderline of living and non-living things. They may show certain properties of living organisms, and they may also show certain properties of non-living organisms. So that's why we have not categorized them into a particular group until now. In the kingdom classification, we see that virus fits nowhere. There is no classification present for virus. Maybe in future we can have some classification, but until now, whatever classification we know, there is no classification present for viruses. Viruses are unclassified until now. So again, if there is any kind of question now, you can ask again, so that
सर वो लास्ट वाला नॉन लिविंग वाला पॉइंट समझ में नहीं आया क्रिस्टलाइजेशन का
mean crystallization क्या होती है? What is crystals? Crystals are geometrical shapes. Okay, they are geometrical shapes, like for example the crystal of sugar, the crystal of salt, the crystal of copper sulfate, or many other things. So crystals are the properties of non-living things. Non-living things can be crystallized. They can be converted into geometrical shapes, but living things cannot be converted into geometrical shapes. Remember, only if a thing is particle, they can be crystallized. If they are not particle, they cannot be crystallized. So viruses are particles. They are particles, so they can be crystallized. If they were cells, they could not be crystallized then. Okay, like bacteria is a kind of cell, so bacteria we cannot crystallize, but virus is not a cell, so it can be crystallized. Yes, Arisha. Yes, any question now?
Sir, can you re-explain the genetic recombination?
Genetic
And mutation.
recombination, genetic mutation are basically change in the genetic material, change in the chromosome. So virus can make a change in its DNA. It can make a change in its RNA. That's why we call they show mutations. As I give you an example, that a virus present in Pakistan will be different from a virus from the same virus present in China, or present in America, or present in any other country. That is because they have different kinds of mutation in them. Their genetic material factors. Okay, genetic recombination means that they can attack another body, and from them they can take a kind of gene. Okay, so they can attack a bacteria, they can take genes from the bacteria. It means they show genetic recombination. That is only the property of living things. Non-living things cannot show this property. Clear? Meant by non-cellular, Arisha, non-cellular means they are not cellular organisms. They do not have any cell. Any other question? बैक्टीरिया में होता है जेनेटिक इंजीनियरिंग और जेनेटिक रिकम्बिनेशन। बैक्टीरिया आपस में भी जेनेटिक रिकम्बिनेशन करती है और बैक्टीरिया जो है वो दूसरे ऑर्गेनिजम्स के साथ भी करती है। वी विल डिस्कस दैट इन बैक्टीरिया एज वेल।
मतलब सर वो उसके जीन्स भी वो ले सकती है?
जी, they go into the body and they normally have the genes copied as well.
अच्छा।
Okay, any other? Is it clear? Clear to all of you?
यस सर।
Yes,
Okay.
sir.